All right. So my name is Dr. Constance Bailey. I'm a senior lecturer in the School of Chemistry, and my research focuses on natural product biosynthesis and generating natural products that come from microbes through bioengineering. So as a chemist, I've always been fascinated by these incredibly complex molecules that are made by microorganisms, mostly microorganisms in the soil and sometimes in marine environments. And they have incredible bioactivity. In fact, a really compelling statistic is 70% of our antibiotics come from soil bacteria, so such a rich source of bioactive molecules for discovering new medicines, particularly to sort of combat some intractable problems. One issue that I've been focused on is antimicrobial resistance. So these molecules are made by bacteria and are quite complex, and so making them synthetically is often quite challenging. And so what I've always been interested in is understanding the chemistry that that life uses to make these complex molecules, and then understanding how we can use sort of bioengineering and genetic engineering to get these organisms to do that chemistry for us in a controlled fashion. This is also a very exciting area being in Australia because we have so much biodiversity in this country with this wild range of ecosystems that are across our lands. And so some of my research is focused on how do we look for new molecules that might be due to our geographical uniqueness in the world. And so the research in my lab really focuses on a few different approaches. One is using a technique called heterologous expression, where we take pathways that might not be made efficiently in their natural hosts and put them in genetically tractable hosts. We also have a whole suite of different techniques that we use to optimize this, one being cell-free protein expression, which is a strategy to help us figure out what genetic elements that we use. I also do some enzymology and biocatalysis, looking for new chemical transformations that are done in these organisms. And then genome mining, because these genes are clustered in predictable biosynthetic gene clusters, we can actually sequence genomes, examine them, and look at new molecules that might arise. And we have some projects in my lab focused on this, both looking at terrestrial samples and also some marine samples from the Sydney Harbor. So just to get a couple flavors of some of the projects in my lab, one project focuses on heterologous expression. So bacteria can either be gram-negative or gram-positive, and gram-negative bacteria have this double-barreled outer membrane that is quite impermeable, and indeed many new antibiotics that are discovered don't permeate this outer membrane. So our antibiotic of last resort for gram-negative infections is polymyxin B, which is very powerful and actually quite nephrotoxic. So a lot of the antibiotics that are used to treat gram-positive infections like vancomycin actually don't permeate the outer membrane, and we're always looking for new targets that can permeate the outer membrane. One example is this molecule darabactin A. It binds BAM-A, which is an essential protein in the surface of gram-negative bacteria, and it comes from a symbiont of a nematode, and it has this very promising activity, but because it has this complex cyclic structure, it's difficult to access synthetically and not made in high quantities by the natural organisms. So my lab has sort of devised a novel strategy to make this molecule using synthetic biology, putting it in actually gram-positive hosts where it cannot kill the organism, because E. coli is a gram-negative host, and that's our most common heterologous expression strain, and it's not produced efficiently in E. coli. So we have a whole program on this. Another exciting direction is that there's a halogenated variant that was isolated from the Tasman Sea, sort of showcasing our biodiversity, and we've been able to make it in our heterologous expression system as well as characterize the halogenase. And this enables potential for semi-synthesis going forward to further optimize the activity. So moving beyond darabactin, we've also looked at collaborating with biotech company owner Ern Lacey, who samples soil from all across the continent. We've collaborated with him to find novel biosynthetic gene clusters, and one that was really intrigued us are called lasopeptides, which have this interesting knot. So they basically are a peptide that has this amide bond with this threaded tail, and this is very sterically strained, which creates new topologies that interact with biology in novel ways. And these molecules, so because of this strain, are not accessible synthetically, but also they tend to be produced cryptically. So they're not produced under culture conditions because they're only produced in certain ecological contexts. So my lab has been focusing on identifying novel lasopeptides and producing them through heterologous expression. One that particularly intrigues us is this one with high amount of positive charge, because we think it's likely to stick to biological membranes, perhaps analogous to polymyxin B, which has all this concentrated positive charge, which is responsible for its bioactivity. So those are sorts of projects that we work on in my lab. And with that, I'd like to thank my research group, my many collaborators, both overseas, across Australia, and within the University of Sydney, both in the Faculty of Science, Faculty of Medicine and Health, and Faculty of Engineering. And thank you so much for your attention.